Dear students, myself Amit Patel. I am working as assistant professor at Anand Agriculture University. Today we are going to discuss paper number 7, that is Technology of Milk and Milk Products, in which we are going to discuss module number 18, which is probiotic and symbiotic dairy products. Inappropriate nutrition is recognized as a primary factor in unachieved genetic potential, reduced mental and physical performance, and increased susceptibility to disease. In searching for effective dietary intervention strategies, a new class of food known as functional food has evolved rapidly, and many new terms such as probiotic, prebiotic, nutraceutical and food for specified health use, which is also known as FOSU, have emerged in response. The term functional food originated in Japan in 1980s when it was used by the industry to describe foods fortified with specific ingredients imparting certain health benefits. There are many possible definitions for the term functional food. However, functional food is usually described as food that promotes health beyond the provision of basic nutrition. Probiotic bacteria are live microbial strains that, when applied in adequate doses, beneficially affect the host animal by improving its intestinal microbial balance. Probiotic foods are food products that contain a living probiotic ingredient in an adequate matrix and in sufficient concentration so that after their ingestion the postulated effect is obtained, and it is beyond that of usual nutrient suppliers. The most common probiotic dairy products worldwide are various types of yogurt, other fermented dairy products, for example cultured buttermilk in Finland, various lactic acid drinks such as Yakult, and mixture of probiotic fermented milks and fruit juices. Probiotic cheeses, both fresh and ripened, have also been launched recently. Prebiotics are non-digestible food ingredients that beneficially affect the host by selectively stimulating the growth and or activity of one or a limited number of bacteria in the colon. Prebiotic foods are food products that contain a prebiotic ingredient in an adequate matrix and in sufficient concentration so that after their ingestion the postulated effect is obtained and is beyond that of usual nutrient suppliers. Symbiotics are mixture of pro and prebiotics that beneficially affect the host by improving the survival and implantation of selected live microbial strains in the gastrointestinal tract. Now let us understand the concept of probiotic. The first record of ingestion of live bacteria by humans are over 2000 years old. However, at the beginning of this century, probiotics were first put onto a scientific basis by the work of Metchnikoff at the Pasteur Institute in Paris. Metchnikoff hypothesized that the normal gut microflora could exert adverse effects on the host and that consumption of sour milk reversed this effect. Metchnikoff refined the treatment by using pure cultures of what is now called Lactobacillus delbrueckii subspecies bulgaricus, which with Streptococcus salivarius subspecies thermophilus is used to ferment milk in the production of traditional yogurt. A formal probiotic definition is a live microbial feed supplement which beneficially affect the host animal by improving its intestinal microbial balance. Over the years, many species of microorganisms have been used as probiotics. They consist not only of lactic acid bacteria such as lactobacilli, streptococci, enterococci, lactococci, or bifidobacteria, but it also include bacillus species and fungi such as Saccharomyces species and Aspergillus species. Now, let us see strains used in manufacture of certain probiotic dairy products. The probiotic strains used in manufacture of dairy products mostly belong to Lactobacillus and Bifidobacterium genera. The characteristics of probiotic strains vary and hence each strain require individual study. The primary requirement of a probiotic strain is that it should be adequately identified with methods based on genetics and that the strain should be defined in the text of the product package. This makes it possible to analyze the scientific data behind any claims made. Some probiotic strains are sufficiently proteolytic to grow excellently in milk, but others need growth stimulants. Sometimes the texture of the taste of a milk product fermented with a probiotic does not meet with consumer demand, and hence probiotic strains are used as adjunct with standard starter cultures. Probiotics can be added before the fermentation of milk, or part of the milk can be fermented separately with the probiotic strain and then it is mixed after the fermentation. In this slide, the most common species of bacteria used in manufacture of probiotic dairy products are displayed. They belong to Lactobacillus species, Bifidobacterium species such as Bifidobacterium bifidum, Bifidobacterium infantis, Bifidobacterium brevi, Bifidobacterium animalis, Bifidobacterium adolescentis. In this slide, we have seen that two major species of bacteria are used as probiotic in manufacture of probiotic dairy products. They are Lactobacillus species and Bifidobacterium species. Now, let us discuss health benefits associated with consumption of probiotic foods. Main positive effects associated with probiotics include cholesterol and or triglyceride reduction, anti-tumor properties, protection against gastroenteritis, improved lactose intolerance and stimulation of the immune system through non-pathogenic means. For centuries, folklore has suggested that fermented dairy products containing probiotic cultures are healthful. Recent controlled scientific investigation has supported some of these traditional views, suggesting the value of probiotic as part of a healthy diet. In addition, the emergence of some new public health risks suggest an important role for effective probiotics. Probiotics may be avenue to provide a safe, cost-effective, natural approach that adds a barrier against microbial infection. In the next two subsequent slides, we can see some of the reported benefits of probiotic organisms. Some of the selected health-related attributes of probiotics include physiological effects such as production of bacteriocin, then antagonistic effect against pathogens, other effects like action on the digestive tract which includes enhancement of lactose digestion in lactase deficient people, then prevention of intestinal disturbances, then stabilization of Crohn's disease, etc. One important health related aspect associated with consumption of probiotic food is alteration of the intestinal microflora, which helps in balancing of the intestinal bacteria by colonization. It also helps in decreasing fecal mutagenicity, etc. Action on diarrhea is also one of the health related attributes of probiotics used in fermented milks. Then systematic effects are also associated with consumption of probiotic bacteria, such as they improve immune health, they stimulate phagocytic activity, they are also associated with reduction in risk of various cancers, they also help in reduction of serum cholesterol. We have seen different health related benefits associated with consumption of probiotic foods. Now, it is very important to select a particular strain which will provide health benefits. So, now let us discuss selection criteria for probiotics. In selecting a probiotic microorganism for use in food, the strain must be identified at the genus, species and strain levels. Understanding the complex interaction among microorganisms in the intestinal ecosystem requires method of differentiating a strain of interest from other strains of the same species contained in the indigenous microbiota. According to FAO and WHO guidelines for probiotic use, specific health benefits observed in research employing a specific strain may not be extrapolated to other closely related strains. In human, the most common used vector involves fermented milk products and over the counter freeze dried preparations for lactic acid bacteria in capsules. Recently, the market has expanded to include other foods such as flavored drinks and pharmaceutical preparations such as tablets. Now, let us discuss selection criteria for efficacious probiotics. They must be having some beneficial effect to the consumer or host. They must be non-pathogenic and non-toxic. They must be in sufficient number, that is, it should contain a large number of viable cells. They should be able to survive in the gastrointestinal tract, that is GI tract of human being. They should also give good sensory properties to the final product, and preferably they should be isolated from the same species as the intended use. The theoretical basis for selection of probiotic microorganism includes: first one is safety, they must be safe for the human consumption. The other one is functional behavior such as survival, adherence, colonization, antimicrobial production, immune stimulation, anti-genotoxic activity, etc. And third important aspect, theoretical aspect for selection of probiotic microorganism is its technological aspect, that is whether they are able to grow in milk, whether they are able to provide sufficient or required sensory properties or not, their stability during processing as well as storage, then resistance against phage. All these factors are also to be considered while selecting a probiotic bacteria to be used in manufacture of fermented dairy products. Till now, we have discussed the concept of probiotic. Now, let us understand the concept of prebiotics and symbiotics. A prebiotic is defined as a non-digestible food ingredient that beneficially affects the host by selectively stimulating the growth and or activity of one or a limited number of bacteria in the colon. It is substance which modifies the composition of the colonic microflora in such a way that a few of the potentially health promoting bacteria, especially lactobacilli and bifidobacteria, become predominant in numbers. A symbiotic is the product in which both a probiotic and prebiotic are combined in a single product. It is defined as a mixture of a probiotic and prebiotic that beneficially affect the host by improving the survival and the implantation of live microbial dietary supplements in the GI tract. By selectively stimulating the growth and or by activating the metabolism of one or a limited number of health promoting bacteria. The prebiotics developed so far are the non-digestible oligosaccharides and especially the non-digestible fructans, among which chicory fructans play a major role. Prebiotics have been defined as non-digestible food ingredients that, when consumed in sufficient amounts, selectively stimulate the growth and or activity of one or a limited number of microbes in the colon, resulting in documented health benefits. With probiotics, there is an emphasis on viability and dose consumed. Prebiotics, on the other hand, positively influence selected microbes. Since both types of ingredients have the intestine as functional target, it is obvious that the two can mix and may reinforce each other's effect, thereby creating a symbiotic, a mixture of probiotics and prebiotics that beneficially affect the host by improving the survival and implantation of live microbial dietary supplements is the GI tract by selectively stimulating the growth and or activating the metabolism of one or a limited number of health promoting bacteria and thus improving host welfare. The clue of prebiotic compounds is that they are not digested in the upper GI tract because of the ability of the digestive enzymes to hydrolyze the bond between the monosaccharide units. They act as soluble fibers and are digested in the colon, enhancing microbial activity and stimulating the growth mainly of bifidobacteria and lactobacilli. Such compounds, which are either partially degraded or not degraded by the host and are preferentially utilized by bifidobacteria as a carbon and energy source, are referred to as bifidogenic factor. Some bifidogenic factors of commercial significance include fructooligosaccharides, lactose derivatives such as lactulose, lactitol, galacto-oligosaccharides and soybean oligosaccharides. Consumption of higher doses of prebiotics may encourage the formation of gas, flatulence and intestinal discomfort. The end products in the gut fermentation are mainly short chain fatty acids such as acetic acid, propionic acid and butyric acid, lactic acid, hydrogen, methane and carbon dioxide. Short chain fatty acids, especially butyric acid, are known to act as an energy source for enterocytes. The main dairy products enriched with prebiotics are yogurts and yogurt drinks, but spreads, fresh cheeses and milks are also on the market. Galacto-oligosaccharide, which is milk-based prebiotic, is derived from lactose by the beta-galactosidase enzyme. It is a natural prebiotic from human breast milk and it facilitates the growth of bifidobacteria and lactobacilli in breastfed infants. Galacto-oligosaccharides are commercially used principally in Japan and other parts of Asia. In Europe, inulin and fructooligosaccharides are widely used in various functional foods including dairy-based products. Inulin is a group of fructose polymer which is linked by beta 2,1 bonds that limit their digestion by enzymes in the upper intestine. Their chain lengths range from 2 to 60. Oligofructose is any fructose oligosaccharide containing 2 to 10 monosaccharide units linked with glycosidic linkage. Both inulin and fructooligosaccharides are extracted from plant material, that is chicory, or synthesized from sucrose. The role of inulin and the oligofructose in a food matrix is bifunctional. They do not increase the viscosity of milk product, but they give a richer texture to liquid products and spreads. Now let us understand what are the symbiotic food products. Symbiotic food products means when we combine probiotics and prebiotics in a single food, that product is known as symbiotic food. The expected benefits are as stated in the symbiotic definition, which is an improved survival during the passage of the probiotic bacteria through the upper intestinal tract and a more efficient implantation in the colonic microbiota together with a stimulating effect on the prebiotic on the growth and or the activities of both the exogenous, that is probiotic, and endogenous bacteria, that is bifidogenic bacteria. Symbiotics are much more than just a mixture of pre and probiotics. As the name suggests, a synergy must exist between two components and hence not just any mixture will be a symbiotic. The majority of yogurts marketed in Australia and United States and Europe in recent years contain probiotic bacteria and some form of prebiotics. Such combination products are referred to as symbiotics. Japan is the world leader in probiotics and prebiotics products. A symbiotic is a marriage of concept of probiotics and prebiotics. The advantages are that a commercial probiotic with known benefits can be used and the prebiotic aids the establishment of the organism in the complex colonic environment. The survival of the probiotic part of a symbiotic can be determined in vitro using a model of a human gut. Whereas, the concept of prebiotic has become very popular since its introduction in 1995. The concept of symbiotic has up to now not really been applied to the development of new foods, especially functional foods. Furthermore, even when new products indeed contain a mixture of pro and prebiotics, they are seldom presented as defined as symbiotics. This concept thus remains open for validation and further research is needed. In the conclusion, I would like to say that probiotic bacteria are those bacteria which are when ingested in human guts provide health benefits. The second concept we learned was about prebiotic substances. Prebiotic substances are non-digestible food components which help in the growth of probiotic bacteria. And when we mix these two things together in a manufacture of fermented dairy product, that is probiotic bacteria as well as prebiotic substances, the final product is known as symbiotic dairy product. Thank you.